Dr. K.K. Wong is a professor in the Department of Gynecological Oncology and Reproductive Medicine at MD Anderson Cancer Center. His research is among the projects STAAR has funded. Dr. Wong will be with us to discuss laboratory discovery science and what he is working on in the field of LGSOC. In his leisure time, he enjoys running, and lately he has completed his 16th Houston full marathon. Wow. Dr. K.K. Wong is a molecular biologist with specific training in molecular genetics and genomics, and in DNA sequencing analysis, gene expression profiling, and data mining. He has been working at MD Anderson for almost 20 years. He has published over 110 peer-reviewed articles and 13 book chapters. His major research focus is to decipher the molecular pathogenesis of ovarian cancer with a goal to identify biomarkers and therapeutic targets for individualized therapy. One of his projects is to decipher the resistance of mechanisms of low-grade ovarian carcinoma to MEK inhibitors. He has developed cell line and genetic mouse models for LGSOC cancer study. He is currently applying genomic technologies to get a deeper understanding of the molecular changes in LGSOC from patient samples. So, give Dr. K.K. Wong a big welcome.
Thank you, Brady's introduction, and so special thanks actually to Dr. Gersonson, because he is the one that recruited me to MD Anderson, to work on low-grade ovarian carcinoma. So one of the you asked me about the insulin growth factor, So, right, I think when we published the paper a while back, sometimes I also got email from patients like you, but usually I will refer to Dr. Gersonson because I'm a researcher, right? I cannot give them clinical recommendation. So one of the patients told me, oh, actually, I think she is a nutritionist. He said, you just have to walk and exercise, and that can lower your level of insulin growth factor 1 in your bloodstream. So maybe that can help, But back then, the drug targeting IGF-1R is, I think the drug company said it's too toxic, and then they don't want to sponsor the trial. So what I'm going to do today is, it's like I'm teaching to my graduate student. Okay, so so this picture is, so the top part is how the low-grade carcinoma looks like under the microscope. Some small cell oval shape, small nuclei. And then the middle, good. Just be joyful is good, So the middle part is a mouse that we generated. It's the first mouse that will develop a low-grade carcinoma, look like human tumor. So we can use that to study how the cancer development, right? Because we cannot collect tissue, biopsy from a patient, right? So we can use a mouse to study it and see whether what we learn from the mice can be applicable to a human. Okay. So I will talk about like the basic molecular biology of cancer cell. There is some slide I used in my lecture for the graduate student a long time ago. And then we will talk about, so because initially we want to have a lab tour, but right now there's a lot of restriction in the hospital, so it seems we are not able to visit the lab. And finally, I will talk about the two projects. The STAAR Foundation has supported the lab. In terms of the support, I think Jane also have provided some significant funding to keep the lab sustainable. So basically, what's a cancer? I mean, I was always asked by my friend. I mean, I have a friend that have lung cancer, brain cancer, unfortunately. So what's a cancer? Cancer is a genetic disease. Something change or mutation in our DNA. Yeah. But it can happen, most of them are by chance, sporadically, right? Some, probably maybe 5 to 10% can be hereditary, but most of them just by chance. So what is DNA? So people ask DNA, right? So the cell, if you blow it up 10,000 times, it'll be about the size of your fist. Yeah. So in the cell, there's a nucleus, and each of us has 23 pairs of chromosomes. Right? So the chromosome is like a bow tie. Right? Has a knot, and then has a long arm. We call it P, and then we call it Q, the longer one. So the DNA is wrapping around some protein called histone. Yeah. And we can unwire it, and you can see the DNA. And the DNA has 4 basic components. We call it a base. Yeah, that's important. I mean, remember, there's only 4 components, G, C, A, T. Yeah. But why? And then we call it a base pair, because the DNA is not just a single chain, it's a double helix. What we call it is always in pair. Like the G always pair with the C, A always pair with the T. So how big is the DNA, or how big is the base pair? So you can calculate how many cells we have. Right? We have about, how many? 10 trillion cells in our body. Right? Each of our cells has 3 billion base pair. So you multiply the length of each of the base pair with the number of cells, that is, we have about 20 trillion meter long. So if you can stitch all your DNA, so how long is 20 trillion? Is you can go back and forth from the Earth to the Sun almost 70 times, actually 69.2 times. So that's how amazing this length of DNA can be wrapped in a small, tiny cell. Right? But so the DNA has to be, during the cell to divide, the DNA has to separate, to make a new DNA. But during this process, some mistake can happen, can be caused by some environmental carcinogens or just the body. Somehow the process is not that accurate, so make a mistake. But our body can fix it. But if that mechanism has go wrong, then correcting the mistake is not good, then you got a mutation. So the rationale is from the DNA to RNA to protein. So if you have 40 DNA, then you may have a bad protein. So there are many causes that can cause some DNA changes. So it can be like if you're exposed to UV light, so the UV light will cause the T to link together. As a result, when the cell try to replicate, it will make a mistake. So exposing to the UV light too much will have a high risk of skin cancer, for example. Or some other, like some phenolic compound, they can also will bind to the DNA and cause some mutation. But you have a mutation doesn't mean you have a cancer. Right? It's a natural process. Only if those mutations hit some of the gene. So the cancer, or we call them cancer gene. I will talk about this later. So how we classify the cancer gene into 3 different categories. So besides the change in the DNA, like the base should be A, suddenly become T, or suddenly a piece of DNA is lost, there are some other changes can be affected by what we eat or or other environmental stress. So here I talk about the epigenetics, but I will not touch it too much here. And the gene that will cause cancer, we have two kinds. One mainly is like some is oncogene that can promote the cell to grow. And then, but there are also some anti-oncogene or tumor suppressor. They will block the cell from becoming cancer. So it's kind of the two forces they're fighting each other. So there are many types of mutation. Like, as I told you, there are 4 basic units, right? C, T, A, G. If any one of those become a different one, then the outcome will be a bad protein, or you lost a tumor suppressor, then the cell will become cancer. Or you can somehow lose one of the base, or you can have addition, or you can the chromosome can swap. Yeah, we call it inversion. But this is a smaller scale changes. But there are also like changes at the whole chromosome level, like the whole chromosome deleted, disappear, or some chromosome, like chromosome 19 swap with chromosome 1, for example, or some piece of the chromosome just get an extra copies, right? If that extra copies is in one of the oncogene, then the gene may produce more protein and cause the cell to keep growing. So at the higher level, yeah, this the first the top part. Let me see. Yeah, see here. Yeah, right. This is what we can do called the sky is a technique. You can paint each chromosome in a different pseudo color. So, for example, like each of these is a chromosome, right? Like a bow tie. But for some chromosome, you can see 2 different colors together, which means some region has been swapped with the other chromosome. Yeah, so this is a chromosome aberration. And those are so by using those cytogenetic techniques, you can only just tell you what has happened, like what kind of chromosome has swapped. But we can use, we published a paper looking, using whole genome sequencing. Basically, we sequence the 3 billion base pair from 14 patient sample. Yeah. So in the past, I mean, as Dr. Gerstensen mentioned, in 2003, the first human genome completed close to 92%, and US spent about $13 billion to get the first human genome sequence over almost 14 years. But nowadays, probably, so for this project, it's still expensive. Like one patient in the lab, we can, so right now is $5,000 to $6,000. We can get one patient genome sequence. And by doing that, we can get very precise what has changed in the chromosome, like where is the breakpoint, which piece of DNA is gone, yeah, which region has increased in copy numbers. So from this paper, what we found is one of the regions is on chromosome 9, 9p, is quite frequently deleted. Almost 30% of the patient has that region deleted. So what happened? You lost a piece of DNA, so you cannot make RNA, you cannot make protein. So when patient lost that piece of DNA, the 9p, so they were not able to make a protein called P16. Okay. So P16 is encoded by a gene called CDKN2A. So there is some cell study. So people look at the ovarian cancer cell. So when a cell does not express this P16 protein, they are more likely to be sensitive to CDK4 inhibitor. That is the one that in clinical trial. So we are trying to test whether that is true by looking sample from those patients. So that's why it's important to get as much information from the patient. So I applaud you because patients signed a consent to allow us to analyze their patient sample, right? Those are sample or residual sample after the pathologist make the diagnosis. Yeah. So we talk about the chromosome changes. So about cancer genes, right? So there are three major categories of cancer genes. Right? One is tumor suppressor genes. I just mentioned in low grade, most likely is one of the tumor suppressor genes is the CDKN2A. So when this gene is lost, so normally this gene control like how many times the cell will divide, because all our cell has a limited number of time they can divide. So maybe the hair is losing, maybe the cell is not dividing anymore. So there are some genes control the number of cell cycle. Yeah. And there are some called oncogenes. It's like KRAS or BRAF. Those are the genes that normally they only respond when the cell need to divide. Yeah. But when those gene is mutated, the cell no longer need any signal. Like when you have a wound, then the blood flow over there and stimulate the cell, like some epidermal growth factor or some other growth factor will stimulate the cell to divide and repair the wound. But for cancer cell, when KRAS is mutated, so the cell just keep growing. Yeah, even though there's no signal asking the cell to proliferate or to divide. The third kind of gene is, we call it stability genes. It's gene involved in DNA repair, like the BRCA1, 2, and other MXH2, MXH6. So those genes are some guardian, like, because we constantly have mutation in our body, right? Only if those mutations hit those cancer-related genes, then you may have cancer. Yeah. But during this process, our body also have at least 6 different mechanisms to repair the damage. Any mutation changes, our system will repair it. Yeah. So but if someone has a BRCA mutation, so their repairing mechanism is not good. So one arm of the repairing mechanism is defective. Then the cell just keep accumulating more and more mutation. Yeah, eventually, you hit one of the cancer gene, then cancer will develop. So I just use an analogy, right? Just like, KRAS is mutated. It's like somehow the gas pedal is wrong. So the car just keep going, but the handbrake is still on, right? So I was thinking this may be the borderline, right? Borderline tumor may be the RAS signaling is on, so the cell just keep growing, but there's still some of a force, we call it tumor suppressor, still holding it back, right? So it may not become a cancer. Yeah, I will talk about the differences between the cancer and the borderline. But if both genes got mutated, you have a oncogene like KRAS, or BRAF, then the cell will keep dividing, growing, and then your brake is broken, and then you're just going to be an accident, you're into cancer. So from the study of many different cancer types, so people come up with the cancer has some characteristics, at least 10 different characteristics. One is we talk about, like we start from the proliferating signal. For example, the KRAS is mutated, so now the cell just keep proliferating. So this is the mechanism. We know at least 40 or 60% of the low grade has KRAS mutation. So we know this is one of the mechanisms. So that is the trametinib, trying to block this signal, right? And like evading growth suppression, maybe the P16 is one of the mechanisms. So the CDK4 inhibitor, now trying to see whether it will work in patient. But another area we don't know is, because cancer cells are very smart, normally when some of the cell has some changes or become defective, our immune cell will just clear it out, right? Remove it from our body. Or the cell will suddenly just die by itself, it's no longer working well. So this area we don't have much study, yeah. And some cancer cell will just keep making, it seems it always has inflammation, so that sustains the cell to be there, yeah. And another area is like the differences between the borderline, serous borderline, and the low grade is the cancer cell start to destroying the normal cell, or we call it invasion. So you go inside the normal cell. And the mechanism, we still don't know. Yeah. When you ask about what's the implant is like just some cell, yeah, just stick it to the surface. But the cell hasn't go inside the tissue. It hasn't invaded the normal tissue. Yeah. So I think it's like a chewing gum, you just stick in the wall, unless you punch a hole and then they become a cancer. Yeah. So to do this process, the cancer cell also has to stimulate the body to make some blood vessel, right? We call it angiogenesis. It's right here, inducing angiogenesis to make some new blood vessel into the tumor to provide the nutrient for the tumor to keep growing. Because you cannot keep going without getting blood supply. That may be the reasons some of the patients is benefit from the treatment with bevacizumab, which block the blood vessel growing. Of course, there are some other characteristics. We still don't know the underlying mechanism, how the cell can have this characteristic in low grade. So talk about cell signaling. This is a very important area. Yeah, this is how the cell sends a signal like from the outside environment, like light or in the blood, in the circulation. Yeah. So, and in our body, we have a lot of, we'll make a lot of growth factors, and the cell only need it when you need to divide or grow. So this, but when there's, so here, yeah, it was animated, but now it's not animated, okay. So this is a growth factor. It binds to the receptor. So our cell, surface has a lot of like receptor, basically some protein, like antenna that will sense the signal. And this signal will pass from one protein to another protein, and then to a KRAS protein. So the KRAS is like a switch, right? So only when, normally, the normal KRAS require the signal to turn on the switch. But when this KRAS mutation is mutated, this switch always on, okay? Yeah, so although KRAS mutation has been discovered more than 30 years ago, and up to now, probably there are some new clinical trial, because in the past, so the active form to the inactive form rely a small molecule called the GTP here, yeah. So you bind to the KRAS, and then it switch on the protein and turn on the signaling pathway, and then the cell will proliferate, yeah. So for many years, the pharmaceutical company try to develop an analog, look like the GTP, to block it, yeah. But it's not successful, because normally the GTP is so efficient binding the protein to activate it. But more recently, there are several biotech companies, they try to develop another drug to block the activated mutated protein. Yeah, so there are some clinical trial but not in ovarian cancer. Right now it's on maybe endometrial cancer and pancreatic cancer. Yeah, there's some trial to block there. And also another thing is mutation, like KRAS mutation is not just one kind. Yeah, so so the protein is very long, but the major signal is, so a protein consists of a lot of amino acids. So the building block of proteins are amino acids. Yeah, so we have require 20 essential amino acids. So the most important protein, the amino acid is in the position 12 and 13. So, and this original is a amino acid called glycine. So it can become an aspartic acid or you become a valine. So there's some correlation. If patient with KRAS G12V, we call it from glycine become valine. It seems there's some correlation. Patient with those mutations will be more responsive to the trametinib, unlike the other G12D, but still work. And trametinib is, because once these two proteins mutated, as I just mentioned, this cell will keep going and cause the cell to proliferate. So this is a chain of reaction. So trametinib that some of you has used is to block this signal. So this protein called the MEK protein no longer can activate this protein. So that's how this works. But this is just a simplified version of the cell signaling. In our body or in our cell, the signaling is like a circuit board. So it's composed of circuits, assembled from an array of intercommunicating components. So when we block one of the pathway, the MEK pathway by the trametinib, we don't know what other pathway will be activated. So we are able to identify those pathways, maybe some kind of combination treatment or some other treatment or sequential treatment may be possible. But this is very complicated. It's not the whole picture also. Our body has about 20 to 30,000 genes. So, and over 1,000 phosphorylated protein. So over the years, since I work on the low grade, I try to summarize what we have discovered so far. So first is, where does the low grade start? Still very controversial. Some people think maybe it's from the fallopian tube, or some people think maybe some cell trapped in the ovary and then become a cyst and then become a benign adenoma. So still controversial. But if all of them, but we still don't believe all of them are from fallopian tube. But for sure, they are from some cyst within the ovary, or cyst adenoma, or some early stage inclusion cyst in the ovary. So this picture is the borderline tumor. So you can tell why they call it borderline. So the darkly stained cells are the cancer cell, right? So they are in the border, and the middle part actually is the normal cell. So it's in like the cyst, and then some cells coming out. So the normal cell is inside. The cancer cell is just in the border, so it's borderline. So we found that some of the pathway like KRAS mutation, BRAF, and some of the ERB2 signaling is deactivated or deregulated. And then during the progression, we characterize some of the low-grade and borderline tumor. We found some of the genes like PAX2 and AGL3, and KRAS mainly, and MRAS will be found in those later stage borderline tumor. And there's some literature looking at like how the patient doing after. In some center, they follow the patient very long, like up to 36 years. Very impressive. So it seems for late stage borderline, about 20%, they will recur as borderline. So when they recur borderline, I think it's okay, right? But still it's borderline. So but for some of them, they will recur as low grade. So if you look at, even this is, although the cancer cell is proliferating, but the cancer cell is stay at the borderline, at the outside. So the cancer cell did not invade or go inside the normal cell. But when it become a low grade, you can tell all the normal structure got destroyed. So all the tumor cells just grow inside. Yeah. So, and we analyze a lot of those patient sample. Yeah, not a lot, but a decent amount of cancer cells. So those are some new mutations that we have identified, like the DNM3, UBR5, and we also look at, we sequence the RNA. We literally sequence tens of millions of RNA molecules in the tissue. Right? And from those RNA, we can tell this RNA is from gene A, this RNA from gene B, so we can construct computationally what kind of pathway is deregulated. So here are some of the pathway we think is deregulated in low-grade serous carcinoma. Yeah. Like the mTOR pathway, I think STAAR also funding Oklahoma to work on the mTOR pathway, see whether targeting mTOR may benefit the patient. Yeah. And the IGF-1 I just mentioned, yeah, we also found that one, but not able to further test it. And another thing is, I think is new is, so ubiquitination is a process. So when the cell make the protein, so when the protein is not necessary, so the cell will degrade the protein, recycle the raw material. So if that process is deregulated, then some protein just keep staying there. So, yeah. And we also identify some region, chromosome region, like chromosome 1P, 6Q, 9P. I mentioned 9P probably has the CDK4, N2A, yeah, the tumor suppressor gene. So when a piece of chromosome is gone, it's likely a tumor suppressor gene is there. But whether that has any, so how can we target those will be challenging, yeah. But we know where to look for them, though. I think many of you probably have done the molecular testing, so you cannot read this, but I think you have the information. Yeah. So, yeah, I mean, if you treated at MD Anderson, they do the molecular testing, you look at, basically, they sequence about 600 genes, cancer-related genes. Why that is important? For example, if you have KRAS mutation, then you can do a trametinib. Right? If you have a BRAF V600, then you can do the first, yeah. So those are targeted drugs, yeah. Especially the BRAF V600. So the protein is mutated. So only the cancer cell has the mutated protein. So the drug only targets the mutated form and will not target the normal form in the normal cell. So that is the beauty of the targeted therapy. But unfortunately, most of the targeted therapy, over time, the cell will somehow evolve and some other pathway will kick on and affecting the growth of the cell. Yeah, this is just a simple, I took some picture around the lab, and those are some of the, so patient sample is very important. So we collect a patient sample, we can dissociate the cells. Yeah, we can make some, some cell is live cell, we can save them. Yeah, we can grow them, but not many times. Yeah, some cancer cell, we can grow them and become immortal. Then we call those cell line. Yeah, right now, I think low-grade cell line probably is limited. Yeah, unlike high-grade cell line, many tens, yeah. But low-grade cell line, probably we have maybe around 12 cell lines, yeah. And I'm happy, recently we also developed one cell line from one of the patients treated at MD Anderson. Yeah, so and then you can also, some extra tissue cancer patient sample, you can put it in the wax. I think I bought one. You can trap it here. Okay, so this is a patient sample. You can trap it in the wax, and then you can cut it in very thin slice, and then you can analyze what kind of protein is expressed in the tissue. Yeah, you can validate them. And here is all the liquid nitrogen tank. So they have liquid nitrogen. We store all the live cell there. Yeah, you know, liquid nitrogen is very cold, minus 196 degrees, right? But the cell or live cell, you can store there basically indefinitely. Yeah. So this is, you look at the microscope, you can see the cancer cell, you can stain it, yeah, like expression. And you can culture the cell in the hood. Yeah, this is a control chamber. We call it a CO2 incubator. You can grow the cell there. Yeah, and then you can amplify. Like you can make more of the DNA, and then you can analyze what kind of mutation or whether that region has changed. Or you can also look at the protein. This is the cell line we actually generated. So this is from one of the patients. So we have been growing this cell in the culture hood for more than a year, almost 2 years now. And we have done some preliminary analysis. So this cell line has no KRAS or BRAF mutation. So it will be a good model to study patients who do not have the mutation. Right? Other cell line has a KRAS, like the 2 cell line I have been using, has a KRAS mutation. Yeah. So this is the culture dish. And then this is the one, the mouse that we generated. So this mouse, like this is, unlike human, yeah, you do know, yeah. So this is the mouse ovary, 2 ovary, and the uterus here. But it has a long horn, so that may be the mouse. Basically, you put 2 mice together, 21 days, then they will have baby. Yeah, they will have 8 to 10, yeah, right? And then after about 6 weeks old, you can put them together, they will start mating again. So we are able to, like genetically, we can disable one of the tumor suppressor gene. So we disable P10, yeah. And then we can activate an oncogene, yeah. So in this mouse, then will develop ovarian cancer. This is a low grade, look like low grade, has a borderline feature, and then the invasion low grade tumor cell here, yeah. But it takes a long time to generate this mouse model, like 3 or 4 years to do it. I mean, the reason is, it's too complicated. Yeah, basically is, like because we have 2 pairs of chromosomes, right? So if you want to delete a gene, you have to delete the gene in one chromosome, and then also delete a gene in another chromosome. So every time you put a male and female, so they only exchange one of the chromosome, right? So you have to make it back twice, and by doing that, take about 20 weeks, yeah. So this is just for one mutation, right? You put in P10, and then you have to put in another one, the KRAS, okay. So why animal model or patient sample is important? Because cancer cell is just not cancer cell, right? A tumor is not all composed by cancer cell. Just by cancer cell, I think we can kill it, yeah. But the tumor or the cancer is a mixture of different cells, right? Like here, it has the cancer cell, and then there's some cell we don't know, we call it cancer stem cell. Like maybe chemo can kill all those fast-growing cells, but the cancer stem cell grows slower, yeah. So fast-growing cells, we can kill them with chemo. But there are also other mechanisms, like low grade. One of the possibilities, other than how fast they grow, low grade also express a protein called castorin. Yeah. Castorin is a protein that can prevent the platinum drug to go inside the cell. Yeah. So we know that low grade has higher level of expression of castorin than high grade serous. So other than that, also have like blood vessel. So the blood vessel, then we can hit it with bevacizumab. Right? They are autoimmune cell. Yeah, so there are other cells. I mean, my thinking is like the RAM 301 trial. So they are trying to, like using the dual inhibitor affecting the signaling. I just talked about the KRAS, BRAF. So they inhibit 2 steps there. And then they plus another called FAK inhibitor. Yeah, I think, but may not be true, is the FAK inhibitor actually targeting other cell type within the structure, yeah, or both. Okay, so finally, I talk about the goal, the two projects that have been supported by STAAR Foundation. So one of the project is to ask the question, so as we mentioned, almost 95% of low-grade serous carcinoma express estrogen receptor. So for breast cancer, if they are ER positive, then almost like 70%, the tumor will respond. I think Dr. Gerstensen mentioned when the patient responds, means the tumor can be partial response or complete response, so the tumor shrink at least more than 30% or completely gone. But why that is not the case in low grade, right? So that is the question I would like to ask. Like from some clinical trial, I mean, the response rate in low grade is less than 14%. So can we predict who will respond to those endocrine therapy or who will not? So, And then the second project is the traumatism. I think one of the patients asked, right, if how they can become resistant. That is another thing we want to study. So before I talk about the estrogen therapy, so we need to understand how the therapy works. Okay, so estrogen is like a growth signal. So you have to bind to the estrogen receptor. It's like the first figure here. So the estrogen bind to the estrogen receptor and cause this protein to form with another same protein. And then this protein will go to from the cell surface, go inside the nucleus, and then we turn on, ask the DNA to make a lot of RNA, specific RNA, only specific region. And then those RNA will make protein, and then the protein will help the cell to grow, keep growing, right? So the assumption is, if the cell expresses estrogen receptor, then this pathway must be activated. If we block this pathway, we should stop the cell from growing or slow it down. But so that's the reason, because this requires the receptor and the estrogen, or call it estradiol, the name of the estrogen. So one way to stop the synthesis of estrogen is because the body makes the estrogen, but require a lot of biochemical reaction. So one of the reactions require enzyme called aromatase. So like lactosol is an inhibitor, inhibit the aromatase, so that the cell will make less estrogen. So although the estrogen can be made from fat cell, mostly from the ovary, right? So that is how the lactosol work. But like the tamoxifen is different. Tamoxifen is a molecule very similar to the estrogen, so that it kind of competing with the estrogen, but it has a higher affinity, so you bind to the receptor, like here. This is a tamoxifen molecule, but when you bind to the estrogen receptor, it will not activate both the DNA binding and then, so you will not activate the protein properly, so that even the receptor bound to the DNA to turn on the signal, it turn on only some of the signal, not able to fully turn on the signal required for the cell to proliferate. So that's how the tamoxifen and works. And finally is the fulvestrant. Fulvestrant is different. So it also binds to the receptor, bind to a different way, but it will cause the receptor undergo some degradation, trigger another pathway to destroy the receptor. So you have no estrogen receptor, of course the downstream signal will all shut off, right? So my hypothesis is probably for some low grade or most low grade, maybe both pathways are operating. Some receptor may not require the estrogen. So we don't know yet. I think the fulvestrant probably will give better result than the other two, but maybe from the study ongoing, maybe we can get an answer. Or I think Dr. Gerstensen mentioned one time maybe we can combine 2 hormone therapy together. Or I think Dr. Sun is also looking at the patient who have been treated by multiple hormonal therapy and see whether we can get a clue this is something feasible. So, recently we published a paper. So to address a question, can we classify patient, ER positive patient, so patient express ER receptor, estrogen receptor. So can we classify them? Most likely will respond, very unlikely will respond. So that maybe some patients we don't need to treat them. If they are not going to respond, then they don't need to have the side effect. So, so our approach is we look at some public data. So there are, we are using breast cancer patient sample because breast cancer, you have thousands of patients that the RNA, millions of RNA inside the breast cancer cell has been done. Yeah. So those data, we call it is a big effort for the past probably 10 years, maybe 15 years ago. So the government has a project called TCGA, the Genome Project. So there are thousands of ER-positive women breast cancer. They know like the 20,000 genes, how many like within the cell, how many gene A and how many gene B is being produced by the cell. Okay, and we also know at least 200 of those genes will be activated by the estrogen receptor. So when the estrogen bind to the receptor, then we'll turn on those 200 genes. So we used one set of over 1,000 patient sample of the breast cancer, and we classify them into 4 groups based on the level of estrogen, RNA, and then the pattern of the 200 genes. So we got some kind of machine learning using logistic regression. So we are able to classify the breast cancer, ER-positive breast cancer, into 4 groups. Yeah. And, and then we use this model and predict another group from UK. They also have done about 1,000 breast cancer patients. And we are able to also group them into 4 groups nicely. And those with the certain pattern, high level of estrogen receptor expression, and a good patterns of the 200 genes, those are patients that live the longest. Yeah. On the other hand, low level of estrogen expression and the poor pattern does not look like the 200 gene pattern. They did poorly. So, but we don't have enough low-grade, what we call RNA profile, to do the modeling in low-grade, so that we can predict, come up with a machine learning model to predict who will likely benefit from the hormonal therapy. But there's a hope, I will talk about later. So we also found some gene, like 6 genes, like FA2, INVA, MALL. These 6 genes, if the cell in breast cancer, if those cells express this gene, it's most likely they are pretty resistant to hormonal therapy, like letrozole, tamoxifen. So I think the future also depends on this clinical trial. So I think Dr. Gerstensen mentioned this clinical trial, right, will be completed end of this year. So they have the target accrual is so they will have like 450 patients taking this clinical trial, right? And comparing chemotherapy versus just letrozole. So up to since end of last year, 418 patients already in this clinical trial. So hopefully in the next 2 years or so, we might be able to get some of the tissue, and then we can do the RNA-seq analysis. And we can model, but the outcome probably will take a little bit even longer, to get whether they are responder, non-responder, and then come up with the algorithm to predict to classify the patient into 4 groups that we have done for the breast cancer. Yeah, another complicated thing about the estrogen receptor signaling is, so I talk about DNA, RNA to protein, right? So the gene or the DNA is not 1-to-1. So one gene can make different length of RNA. Yeah, so we call it a variant or transcript. So from all this, probably this is like 30 years of study looking at all the cells. Yeah, there are at least 24 different kinds of mRNA from the estrogen gene, estrogen receptor 1 gene, or estrogen receptor alpha. And so each of this line is the length of the mRNA. Yeah, some are longer, some are shorter. Yeah, they are the same. Yeah, they are similar here in the middle, but some are shorter. Yeah, and then all this mRNA will collectively can make at least 4 different sizes of protein. So this is the most common one. Has the in the previous figure, it has the activation domain A and 2, so that can turn on all the 200 genes. But there's another smaller protein called the variant 7. So in breast cancer, if a patient's tumor expresses both the normal and the short one, then they will be resistant to tamoxifen. One of the reasons is if the patient has this smaller estrogen receptor, the variant 7, and this estrogen receptor no longer requires the estrogen, but this small protein can activate another pathway, the epidermal growth factor pathway, to cause the cell to divide. Yeah. So let me see. So the current technology, so when we talk about next-gen sequencing, right? So basically we have 3 billion base pair of DNA, right? Or the 23 pair of chromosome of different length. So the first step, you have to shear them into smaller piece, around 300 base pair. Because, and then you have millions of, or even trillions of DNA, small fragments. And then the machine, this machine, what we call short read, then you can read the sequence, like from the end, but you can only read up to like 75 base pair. Yeah. And it's keep improving. Yeah, originally only 30, and then 50, and then 75, now 100, and then 150. And now they claim maybe up to 250. So if you remember, the transcript is like pretty long, yeah. So, and then those are fragmented. So you don't know which transcript is being made by the cell. Yeah, that is the limitation. But you can tell, because when you have a short sequence, like 75 base pair, this sequence can align this one, this one, this one. So you don't know which transcript. So computationally, we just randomly assign it. Yeah, if a piece of fragment or sequence map to multiple, those transcripts, we just assign it to variant 1 or variant 2. So we don't know. Yeah, like we don't know how many variant 7 is expressed by the cancer cell. If we know, maybe we can predict those patients probably has activated epidermal growth factor pathway. Yeah. But there is some newer machine. Yeah, it's from UK. It's called nanopore, and this machine is a newer technology. Yeah, so instead of shearing the DNA into small pieces and read it, and this machine can the intact, like you don't have to shear it, so you can read all the way. Okay, yeah, you can read through the whole thing. I think it's pretty amazing. Yeah, and some paper published, they can read up to like 2 million base pair. Yeah, so this is very impressive. So by doing that, we can distinguish whether this particular transcript is being made by the low grade or not. So we test it so far only tested in one sample, the low grade patient sample, and we surely find that patient has both the longest one and the short variant 7. So that patient probably had activated EGFR pathway. And in some recent paper by Dr. Gert's group, they think some kind of combination with EGFR may be useful for low grade. So this is on the estrogen receptor signaling that we are working on. Another project also supported by the STAAR is we try to understand why the cell will become resistant to trametinib. Right? So the approach we have two at that time. So we have two cell lines, human cell lines. So derived from patient. Both of these cell lines has KRAS mutation. So like we treat them at 2 nanomolar of the trametinib. So like most of the cell will die, but some cells survive. So we just keep treating it, and then those cells originally is kind of stable, not growing like stable disease, right? So we treat them, and then suddenly some of the cells start growing. Okay, so after 6 or 9 months, so we are able to generate 2 of those pair of cell lines. The original cell, the cell has been under selection pressure, in the presence of trametinib. We just keep increasing the concentration. If you start growing at 2 nanomole, it will double it, 4 nanomole. Then some of the cells start growing, and then 8 nanomole. We got 2 cell lines. And then we look at what are the protein changes, comparing the original cell line and the adaptive resistant cell line. So we found several proteins that are defensively expressed. And we also look at the gene, but I haven't really dig into to analyze the 10,000 genes. So 2 of the proteins we are interested. One is called the B7H4. The other is the SERPINE1. So I submitted the paper, but did some revision, and then they don't like it. So I'm trying to address their comment. Basically, we have the cell line. So the major argument is cell line not necessarily is the same as human. Right? I agree, right? So, but we cannot get patient sample after they are treated with trametinib. If you are treated and then you recur, unless Gerson do a secondary surgery and get some sample and they can test it. But that was too invasive. So we have to come up with some other way to address that. So we also did some mouse experiment. We treat the mice, and the mice actually live longer with trametinib treatment because they have KRAS mutation. So the mice, this is the survival curve. Like the mice just treated with some saline, and versus mice treated. So those mice with low-grade serous carcinoma treated with trametinib, they live longer. About I don't know, about 15 days longer. And we also found that when the mice progress, the disease progress, and then in their blood, the SERPINE1 also increased. So another way is maybe we can collect some blood sample from patient on the trametinib treatment before and after, and see whether that is true. And see whether we can target the SERPINE1 pathway. Okay. So with the cell line, we also have done some high throughput drug screening. So we have 2 pairs of cell line, right? The sensitive and the resistant one, and we, in collaboration with Texas A&M, they have the machine. The machine can is a small plate. You can have 384 wells, and then each well you can put like 200 or 300 cancer cells. And then they have another machine that can dispense drug onto each of the cells. And then they sting the cell. The stinging is reflecting how many cells are growing. Then they get a result output from it. So we have data for about for 6 of the cancer cell line, but still too many data for me to analyze. I got something interesting because when they do the assay, they only used 3 different concentrations. So I had to go back and see, For example, one pair of cell line, I have about 1,000 drugs can kill the resistant cell more preferentially than the sensitive cell. But for the other pair of cell line, I only have maybe about 100 drugs that can kill the resistant cells. Yeah. So here are the list of some of the drugs that we have found that, like this is the original cell line sensitive one. The lower the number means the cells are more sensitive to the drug, right? So these are two of the resistant cell lines that we have developed. Yeah. So, but some of the differences is not that drastic. So one of the comments when we submit the paper said, the differences is not that drastic. So it may kill the resistant cell, but it's not, maybe clinically, it's not that beneficial. So maybe some kind of combination that I haven't tested. Yeah. And some of the highlighted gene is involved in the epigenetics. So I think the adaptive resistance also can involve some epigenetics. And there's another drug that can target that area. So yeah, here are some of the paper we published and some of the abstract we presented at the American Association for Cancer Research. Yeah. So there are a lot of things we want to do, but I think one of the things I would like to see if we can classify all the low-grade patients into the best responder to hormonal therapy versus those probably will not be. Yeah. And then for the traumatic resistance, I think we are working with Dr. Gertie's group. We are looking at like the downstream signaling, the phospho-ERK, whether that can predict whether the patient will be responding to traumatic or not. Yeah, I think they submitted the paper, to us. And we have lots of drugs we can test, so that we rely on our clinical colleague, whether some of the drug is available or pharma will support it, or the toxicity what they know about, And then one thing that I think I can do now is the long-read sequencing. So long-read sequencing using the new machine, we can read through the whole thing, right? Another good thing about you're able to read the whole RNA is because I just mentioned before, right? The chromosome can swap. There's some changes. There's some big deletion. They can bring two sequences together. So there's some changes in the sequence. As a result, there's a chance the cell will make some new protein, or we call it neoantigen. So the protein only exists in the cancer cell. So if we can identify those protein, then there's a way to develop drug to only target those protein. So all this study is, are we able to find the needle in the haystack that only the cancer cell express it? So I think this technology has a chance. Right. So at the end, I thank all of you. And these are the two people working the lab. And of course, without Gerson, I won't be here. Without all of you, I cannot do anything. Okay, thank you.